Okay, so for electron transport chain, we will see that so we are transporting an electron and there is a chain. What is that chain? One person is going to take that electron and pass it over to the next. The other person is going to take that electron and pass it over to the next. This is what will happen from the inside to the outside of the cell, or vice versa. Like that is the electron transport chain. So there are multiple nodes and it is energetically favorable from one step to the other. What that means is, once the electron wants to come to this particular gate, because it is energetically favorable, it is energetically even more favorable to go to the next. It is energetically even more favorable to go to the next, and so on. So this is the electron transport chain. So there is a energy that is getting dropped, so it is becoming favorable and it is getting transferred from one gate to the other, right? Now, this is called the electron transport chain. Who is going to be the final custodian of these electrons? The oxygen, right, for us. So this is the final electron. So these all will be there. So there will be electron acceptors. But there is one person who is sitting in the end who is going to be the final custodian of the electrons. That is the oxygen. Okay. There could be others, but yeah. So in here is an example of an electron transport chain, in which you see here that this is the place where it starts. So there is an electron donor, right? So your food, I deleted it. So there is this food that is the electron donor, right? And the electron acceptor is sitting here. So there will be different steps involved that will get this electron and pass it to the next. So goes from protein complex 1, which is gate 1, goes to protein complex 2, gate 2. Gate 2 then transfers electrons to gate 3, then transfers electrons to gate 4, and so on and so forth. And finally, you will get this electron pair. So this is the final. And in this electron transfer, you are seeing that at each step, an H plus is getting released out. So when you are getting this releasing, so you are releasing one H out, the other H out, the other H out. So you can have multiple H pluses going out. What is that going to do to the pH of the cell outside? Reduce that pH. So it is making it more acidic. There is also an electron gradient that is happening in here. So this is a proton, right? So there is a there is a lot of proton on the outside. There is a lot less proton on the inside. So you can imagine that when there is a lot of water pressure on one side, water pressure on the other side, or you have a potential energy barrier. So you have a higher potential energy here and lower potential energy here. What happens? These things the thing in here will actually fall back to the lower potential, right? It's not very complicated idea. Energy is dropping. Similarly, what will happen in this proton? So in here, when you are creating a lot of proton on this side versus a proton on this side, there is a proton gradient. Can you understand that? So you're creating a lot of protons on one side. Now that proton gradient is causing two things: acidity gradient and a charge gradient. Both these things are being caused because of that proton gradient. Understood? It's clear? So that proton gradient will also help us to understand this electron transport. But imagine that this electron is going through these different gates, and at each gate you are moving out these H pluses, and finally there will be a process, there will be a gate called the ATP synthase. This is an enzyme, right? This is a catalyst, this is a protein. So like this ATP synthase is going to use this H plus gradient. You're storing water at one place, you're storing all that water in one place, and then just releasing that water to come down. Because of that energy, you can move some turbines. Similar. So you are creating this water, instead of the water, you are causing this H plus turbine or proton motive force. It's not the potential energy, the proton motive force. Because of that proton motive force, there will be a time where all these H plus will come rushing down. And when it does that, it will do something. What will it do? It will convert the ADP into ATP. This is how you are converting the energy. This is how the electron transport chain is linked to the ATP formation. Okay, so this is the proton motive force, and this entire process we'll see how this is chemiosmosis. What is osmosis? Higher concentration to lower concentration, the movement of solvent, solutes and all that. Actually, it's not the solvent, it's the solutes that move, right? In here, you will see that the chemiosmosis is what is happening. So you are creating this H plus concentration on one side, and then that H plus concentration moves. And you can actually see that proton motive force you can actually calculate using this. So I'm not going into that because this doesn't, we are not going to do research on this in this class. So just know that there is a proton motive force that actually drives these processes. And when we were talking about this ATP synthase, imagine that this is kind of a gate in which all these proton has been accumulating on the outside. Now, because of that proton gradient, what this will do is, imagine that this is a shaft. Okay, and there's a lot of proton that is surrounding outside the membrane. There will be a huge rush in which all these 4 protons will come in the inside. Because of that rush, this entire thing will get rotated. Just imagine like a, what do you call this, gear system, in which because of that flow, you're kind of you're helping that movement of gears. So that is what is happening with these protons. When this things gets rotated, there is these alpha, beta and gamma units that are there in here of the ATP synthase molecule. What this does is it can orient the ADP and the phosphate in a particular orientation because of this entire thing. When you get these things in a particular orientation, these two tend to combine. And that ADP plus phosphate, when they come together in a particular orientation, they tend to combine, and then they produce this ATP. This is a very simplistic, it's very complicated, but simplistically, this is how one of the processes can actually be. You can see one of these processes. Okay? Now, when you saw these different complexes, we have seen that many of these complexes also have iron compounds like Fe2S2 or Fe4S4 and things like that. Now, there could be different kinds of these complexes. Now, I'm not an expert on these, so I'll just read it out. So hydrogenases, flavoproteins, and iron sulfur proteins. So all these different protein complexes, there could be all these complexes. And some of these complexes can have iron and sulfur molecules. So Fe2S2, Fe4S4. So these are called ferrodoxins. Now these ferrodoxins, we can think of them because iron is there, sulfur is there. And these are old processes. We can see these things happening in different kinds of organisms. Maybe the origin of life, when these things were happening, maybe it was an iron sulfur rich system. Could be, because you were trying to use anything that was available to you, and you use the iron and the sulfur. And these two things are available where? Hydrothermal vents. Right? You have the iron, you have the sulfur. So that is another kind of support to the idea that the origin of life happened in these hydrothermal systems, because these very primitive pathways of electron transport and energy generation relied on the iron and sulfur ferrodoxins. Okay, so this is a complicated process, but I am taking it here and making it like very simplistic, in which there is this cell membrane, this is the outside of the cell membrane, this is the inside. All these protein complexes are pushing out these four protons, and in the finally, because of these proton concentrated on the outside, there is a proton gradient. That proton gradient is generating this proton motive force, PMF, and that proton motive force, and this is what we are trying to do, is taking all those protons and bringing it in using that ATP synthase protein complex, and that is actually producing the ATPs. The names of these enzymes are most often related to the job that they do. So in here it is synthesizing ATP, that's why we name it the ATP synthase. Okay, we'll talk about the electron tower in the next class.